All right, we are doing our DNA structure notes today, so if you don't have this paper, go ahead and find it. It's double-sided. You need this paper. Once you got it, let's jump in and get started here. I'm going to make my face a little bit smaller. So DNA structure is what we're talking about today. These first couple slides are not on your notes. They're just sort of to get our minds in the right place. So what do you think? True or false? And by the way, this is sort of more fun when we're having the discussion as a class, but whatever. We're still doing it anyway. So what do you think? True or false? You have 10 trillion cells in your body, and each one contains all of the instructions for making you. What do you think? Is that true or is that false? Let me get my little pointer out here. There's my laser pointer. What do you think? Is that true or is that false? If you said false, you are correct because you have way more cells than that. You have 75 to 100 trillion cells in your body, but each cell does still contain instructions for making an entire you. Your blood cells are a little bit different. We're not going to get into that, but every single other cell in your body has all the instructions for making you. So if I take a cell out of your face, it has instructions for how to make your stomach. If I take a cell out of your foot, it has instructions for how to make your ear. So all of our cells have all the instructions, which is pretty neat. Next up, what do you think? If you stretched all the DNA in your body out, its length would equal the distance from the Earth to the Sun. So every single cell in your body, all the DNA, Earth to Sun. What do you think? It's false again because it's actually 500 times that distance. So you have a ton of DNA in your body. All right, so let's get to our actual notes. First blanks on your paper here. What is DNA? Its full name is deoxyribonucleic acid. Yes, you have to write that whole thing in there. You're in school. You got to do some writing. Sorry about it. I'm going to go somewhat quickly through this video. If you need to pause occasionally to write things down, do that as often as you need. All right, so its full name is deoxyribonucleic acid. This is double-stranded genetic material, so in the blank on your notes, double-stranded genetic material. It works as instructions for building proteins. If you were to look at a strand of DNA, it sort of has a thing like it unzips like a zipper down the middle. That's why we call it double-stranded. Those are the two strands of it that make it double-stranded. DNA is usually found in the nucleus of cells. Hopefully you remember learning about cells and different cell structures as 7th graders. The nucleus is one of those cell structures you talked about. You maybe called it the brain of the cell or the command center. That is where the DNA usually hangs out. All right, so next up we've got what is a chromosome. Now there's nothing to write down on your notes from this slide, but quickly, earlier we said that if you took all the DNA from your body, it would stretch between Earth and Sun 500 times, but that's a really long distance to try to imagine. This slide kind of puts it into a better perspective that we can imagine. So instead, let's talk about the DNA from just one single cell in your body. If you took the DNA out of that cell and stretched it end to end, it would be about the length of a car. About 3 meters long. So that's a lot of DNA to fit into a tiny little cell that you can't even see with your naked eye. So that's where chromosomes come in. Chromosomes help us to condense our DNA, to pack our DNA into the smallest little package that we can fit into our cells. So what is a chromosome? It is condensed DNA. Usually these are X-shaped. This is more information than you need to know, but this sort of just shows the layers and organization inside of your chromosomes. Honestly, your chromosomes are more organized than I am. I don't know about you, but this is way more organized. So you've got your initial DNA strand over here. Your DNA wraps around these proteins called histones. That wraps itself up into this thing called chromatin. Then we got those things looped over each other into chromatin loops. Then we got them into condensed chromatin loops. And then over here, if I move my face, you'll see that this is a chromosome. This layer, this level of organization is really important because your DNA is important. When your body needs to know the gene for how to make stomach acid or something like that, you need to be able to find that DNA, and you need to be able to find it now. So when your body needs DNA, it needs to be able to say, okay, I know exactly where that DNA is, I know exactly where to access it, and this level of organization here you can see on the screen helps your body to access whatever genes it needs as soon as it needs it. Continuing with our notes, humans have 23 pairs of chromosomes for a total of 46. You probably already know this, but you get half of your chromosomes from mom and the other half from dad. Obviously, your biological mom and dad. Now, the 23rd pair of chromosomes, I'm going to move my face over here. The 23rd pair of chromosomes determines your sex. Now, in my class, when I am going to use the word sex, I'm going to be real blunt with you. We're talking about what you got between your legs. Do you have a penis or do you have a vagina? Now, when we talk about this in my class, sometimes I even make my students say these words out loud because we're not saying wee-wee, we're not saying wiener, we're not saying any of this. We're in science class, we're using the real words. And so, again, when we talk about sex in my class, also we're not talking about gender. Right? Sex and gender are two different things. A lot of you guys know that. And so when I am talking about this 23rd pair that determines your sex, it's literally, were you born with a penis or were you born with a vagina? I know it's real blunt. I know at first when we start talking about this, you're going to giggle a little bit because it's a little bit uncomfortable, but it's what it is. Now, the other important thing I need you to know about that 23rd pair of chromosomes, XY means you would be male. XX means you're female. We'll talk more on the coming slides and what that means, but this part right here is very important. XY is male, XX is female. Now again, I know that this is goofy, but I like to tell my students that the Y chromosome is instructions to make a penis. And if you can remember that, you're going to be good to go. If you have XY, you have a penis. If you have XX, you don't have a penis. It's blunt, but it's as simple as that. So keep that in mind. That'll help you be successful here. So let's look at this. This is the image that you'll see in your notes. This is called a karyotype. You don't need to know that. But basically what we did is we literally took a photograph. At least I'm an old lady. Back in my day, what they would do is they would take a literal photograph of a cell under a microscope when all the chromosomes were assembled like this. And they would take that photograph and they would literally cut with scissors. They would print it out on paper. They would cut with scissors out these different chromosomes and they would match up the chromosomes according to size and also according to like the banding pattern. So you can see here in number 8, there's this dark band here and dark band here and a dark band there and dark band there. And so they were able to say, okay, those are chromosome 8, they match together. So it's like if you've ever played a matching card game. That's what they did with the chromosomes. So that's how we make a karyotype. Now, in this karyotype, you can very clearly see the different sort of types of chromosomes that we talk about. These first group, I'd like you to circle this first group. So all the numbered chromosomes, all the way from 1 to 22, circle all those. And we're going to label those autosomes, not AutoZone, like the place where you go for your oil change and your oil filter, but autosomes. Auto generally means like body, and chromosomes refers to chromosomes. So it's like makes up the major body of your chromosomes. Now these other ones down here, I'm going to move my face again, these other ones down here, as we said before, are your sex chromosomes. So whether you have 2 X chromosomes like this person does, or you have 1 X and 1 Y, those are the different ways that it would show up. What do you, based on what I've told you before, is this person going to be male or female? Remember, I said the Y chromosome is instructions to make a penis. Is there a Y chromosome here? There is not. Empty space there. We have 2 X chromosomes, so this must be female. XX means female. Now, this is generally the way a karyotype, a person's chromosomes would show up. There's different genetic abnormalities that can happen that would result in differing numbers of this, but 99.9% of people have their chromosomes look like this. Now, on the previous page, we looked at a karyotype that was female with 2 X chromosomes. So there's nothing to put in your notes for this karyotype, but I just wanted to show you what an XY or a male karyotype would look like. So again, you'll see the autosomes numbered 1 to 22 over here, and then down here in the bottom corner, you'll see XY. And in this case, look, there's a big chromosome and a little one. Now that big chromosome is the X chromosome, and the Y chromosome is actually just a tiny little thing. So it doesn't literally look like the shape of a Y, but it's smaller than the X chromosome. So this, because we have 1 X chromosome and 1 Y chromosome, this one's going to be a male. So again, nothing in your notes about that. I just wanted to show you what a karyotype of a male would look like, since the other one was a female. So let's keep going with your notes. What is a gene? A gene, at its most basic level, is just a section of DNA that gives instructions for a certain trait. So you have a ton of DNA, but if you just take a little snippet out of that, that might be a gene that would give instructions for a trait. So here's sort of our chromosome. Each one of those little bands we were looking at on there, if you unravel the DNA in that section, that might be a gene, and it might code for some trait, such as having brown eyes or hazel hair. Another DNA structure I need you to know, we're on the back side of the paper now. DNA also has a form called chromatin. We call this, this is a loose, uncondensed form of DNA. We call it the working form of DNA, and it's how DNA spends most of its time. So I like to relate this, I know it's sort of goofy, I like to relate it to a computer charger. If you have a computer charger and you need to pack it into a bag, hopefully you might like wrap up your computer charger so it's in a nice little package so that it stays nice and neat together and it doesn't get all tangled. That's like how your DNA works too. When it needs to travel, when it needs to be packaged up, it ravels itself up into chromosomes. But as soon as we need to use it, as soon as you need to use your computer charger, you need to unravel it. You need it to be loose and uncondensed. And our DNA is the same way. If I need to read the information on a certain segment of my DNA, I need to have it opened up. It can't be all tightly bound up in that chromosome. It needs to be opened up so I can read the information there. So that's why it's important that you know this term, is that chromatin is when we're using the DNA, when we're reading it, when it's open, versus the chromosome is when it's all tightly packaged away. All right, so the next section in your notes just asks you to write down something interesting you learned from my story about the discovery of the structure of DNA. So I'm going to try to go through this story as quickly as possible. All you got to do is write down something interesting that you learned. So these are the 4 people who we generally attribute with discovering the structure of DNA. They were doing their work in the 1940s, 1950s. And it was important that we understood DNA because they knew it would help us to develop better medicines. It might help us to develop better diets. In general, it helps us understand how our bodies, how living things work. And we didn't really know what DNA looked like or how it worked before. And so that's why this team was trying to figure this out. Now, the guys in this team, I believe that this one is Edwin Chargaff. I believe that this is either James Watson or Francis Crick. And this is either James Watson or Francis Crick. Don't quote me on that. But the guys, James Watson and Francis Crick, and sometimes Edwin Chargaff, they were working with physical models. So think like Legos or like Tinker Toys. And they were trying to put the different, because we knew what DNA was sort of made of, we just didn't know how the pieces fit together. So they were using models, and this is the model that you see pictured here. They were using this physical model to try to put the pieces together and see what shape it formed when pieces were put together. And that was how they were trying to solve the problem. What's interesting is that they had the structure very close, except for that they had it sort of inside out. So they still had it in this sort of shape, but it was inside out, and yeah, it was inside out. That's all I got to say about that. Now, Rosalind Franklin, she was working with a technique called X-ray crystallography. Now, if you've ever had an X-ray, and maybe you've broken a arm before or something like that, you might know when they do the X-ray, they line the camera up to X-ray whatever part of your body they want to see, and often they'll put a heavy, like lead vest over the rest of your body. It happens, maybe if you go to the dentist, this happens too, where they put the camera sort of at your mouth and then they put this lead vest over you. And if you're not sure why they do that, it's to protect your body. X-rays are dangerous in large amounts. So when they want to x-ray your arm or they want to x-ray your teeth, they want the x-rays only to be focusing on those areas because they don't want it in your internal organs where things are more delicate. They only want to point these somewhat dangerous rays at places where they need to see and not risk harming something else along the way. Well, back in these days, we didn't really understand quite how dangerous x-rays were, and Rosalind Franklin was working with them all day, every day, and she was sort of almost using like a microscope to look through as well. So she was exposing herself to lots of x-rays, like I said, all day, every day. But in doing that, she was able to take literal pictures of DNA, of the actual DNA spirally shape that we know today. She was able to take actual pictures of it. So one day, James Watson or Francis Crick, I don't remember which one, went to go talk to Rosalind Franklin. They were sort of working on a team together. They went to go talk to Rosalind, but she wasn't in her office at the time. However, she had up on her walls all the pictures that she had been taking. And one of them, she had written like a little sticky note on it that said double helix. The guys with their Tinker toys, they were putting them together, like I said, very close, but not quite right. They didn't quite have that double helix shape. So she took that picture that said double helix. She was able to interpret the areas of light and dark in her photo to understand that the DNA was in a double helix shape. Sure enough, the guys saw that and were like, oh shoot, I know what I'm doing wrong. So they went back to their workshop where they were putting it together with a physical model. They took it all apart, put it back together in a double helix shape, and because of Rosalind Franklin's help, which she unknowingly gave because she wasn't in her office to actually tell him this, he just happened to see her work on the wall. Because of her help, they were able to figure out the structure of DNA. So some people say that they sort of stole her work in that way. Other people say they were working on a team, and she would have shared that information with them eventually. She just hadn't gotten to it at the point. Here's sort of the kicker to this whole story. If you can sort of see, all of these guys have these little gold medallions on, and those little gold medallions are Nobel Prizes for discoveries in science, but you might notice that Rosalind doesn't have one on. Why do you think that is? A lot of students say, well, maybe it's because she was a woman. It's a good guess, but no, it's not because she was a woman. In fact, the reason that she doesn't have one is because they don't award Nobel Prizes after someone has died. What do you think Rosalind Franklin died of? Cancer, from the x-rays that she was working with. Those x-rays were damaging the cells in her body. She ended up dying of cancer as a result of the work that she had been doing over all those years. And so she was literally killed by her work before she was able to receive the Nobel Prize for it, which kind of sucks. We all know and remember Rosalind Franklin for her contributions, so she still is credited with these things, but it sucks that she didn't end up getting the Nobel Prize. In addition, I mean, she was a woman working in science in the 1950s when it was just a hard time to do that. So that's the end of my story. I think Rosalind Franklin is an interesting person. If you ever need to do some sort of research on someone, especially if you're interested in women in science or anything like that, I would recommend doing some more research on her. But there's the end of my story. You can pause me, write down anything interesting you learned about that story before we wrap up the rest of these notes. Okay, so next up we've got nucleotides, which we would also call DNA bases. We call these the building blocks of DNA. Or otherwise we would call them the steps on the ladder. So if you imagine DNA as sort of like a twisted ladder, they're the steps along that twisted ladder. There are 4 types of DNA bases. Maybe I can move my face up to here. We've got A, C, G, and T. You can see those in this diagram right here. You'll see that A always pairs with T and G always pairs with C. And it's if you sort of think of it like, again, maybe sort of like Legos, A and T both have 2 little areas where they want to make a connection. G and C both have 3 areas where they want to make a connection. And the separation between the 2 and the 3 is just a little bit too different, where the 2 can't connect to the 3 and the 3 can't connect to the 2. So if you have the 2's go together and the 3's go together, is basically the reason why. It's chemistry, you know? There's a reason why we do chemistry before we do biology. As far as the pairing goes, it's very important that you remember this. A pairs with T, G pairs with C. This is something that's going to come up a number of times in 8th grade. It's going to come up in high school. The way I like to remember it is that the letters A and T are both made up of straight lines. So that's how I remember that they go together. And then G and C both have this big curvy shape to them. And that's how I remember that they go together. That's my memory trick. Some students like to remember that A and T spell the word at, and then the leftover letters just also go together. Whatever works best for you, I don't care, as long as you remember. Now let's put that into practice. So if A always pairs with T, the letter that would fill in this section of the DNA strand would be T. Okay? Take a second, pause me, fill in the rest of your DNA strand. We'll go through it together to make sure you get it right. All right, here we go. Hopefully you paused and filled it in. So we've got T, then A, then C, C, G, A, T, come on, T, and G. So using those pairing rules, if we're given one side of the DNA piece, we're able to figure out the other side of the DNA piece. Again, this is where that double-stranded idea comes in. We've got 2 strands that pair together in the middle. All right, a quick review. If you are able to answer these questions, that means you're good for today. So where do we find DNA in the cell? If you said in the nucleus, great job. What are those X-shaped structures called? Chromosomes. Good job. I heard you say that. Chromosomes. Great job. What are chromosomes made of? Don't think too hard. What's coiled up inside of them? DNA. You got it. Great job. What is the loose, uncondensed form of DNA called? Close. You pronounced it wrong, but chromatin. I'm just making this up. You know that, right? What is a gene? That was written down on your notes. A gene is a section of DNA. It gives instructions for a trait. Section of DNA gives instructions for a trait. Those are important pieces to that definition. Building blocks of DNA, you can either use the big fancy science word or you can use a simpler word. Nucleotides or DNA bases are what we would call those. Those 4 DNA bases, what are those 4 letters? A, C, G, and T. A pairs with and G pairs with. If you said A, T, and G, and C, you're good to go. All right, there's the end of our notes. Thanks for taking them. Let me know if you have any questions.